will introduce myself. I am Nicole Trefault from Universidad Mayor of Chile, and I will present you on with this presentation. You can see this, but this is now the...
That's fine, perfect.
Okay, thank you very much. I am very happy to present now, and I want to thank also the organizers for inviting me to this important symposium. Well, my talk is named Understanding Sponge Microorganisms, Symbiosis in Extreme Environments, so I will change a little bit topic from human to extreme environment and from human host to the most primitive animal. So let me say first that Antarctica is a unique and extreme ecosystem. We have several characteristics here in this continent that make this system a very special one. Antarctica is, for example, the driest continent on the Earth, with around an average of 166 millimeters of rainfall per year on average. It is also the place where the lowest temperature has been recorded, almost minus 90 Celsius degrees, but the average temperature normally is between 1 and 3 Celsius degrees. And it's also the windiest continent with a slow roughage of winds that make this continent to be the windiest from all the continents. Antarctica also contains the 85% of all the glacial ice that exists in the planet. That means that this space, this ecosystem, conserves 77% of all the available fresh water that we have in the world. This striking component or characteristic in Antarctica put a special aspect in terms of conservation and preservation of the resources and all the climatic effect that can have the loss of ice. And this is because when the Earth can reflect only between 15 and 25% of all the radiation that impact the surface of the Earth, Antarctica can reflect almost 90%. That means that most of the heat that impact the surface of the Earth is in fact reflecting by Antarctica and by the white ice coverage. If we think about on whom we depend all the aspects for fauna in Antarctica, we recognize that when in other planets we have significant influence on a terrestrial ecosystem for input of energy and carbon, in Antarctica, this iconic fauna in reality depends almost exclusively on microbial components. And this is because another characteristic of this ecosystem is precisely the strong seasonal variability in terms of primary productivity. Here I show you at the left a picture depicting the amount of chlorophyll that can be recorded during summer, in which you can see most of the colors are green or yellow, while in winter, in the austral winter, that is in July, we have almost completely ice coverage that impact greatly the input of the amount of photosynthetic activity, and by this reason, the amount of chlorophyll, and that means food for the rest of the ecosystem. So, with this kind of things in mind, we recognize also that while climate change is not affecting all the areas of the world with a similar average of impact, we have some places in Antarctica where the temperature increase and the loss of Antarctic sea ice has been more profound, more important than in other areas, particularly the Antarctic Peninsula that you can see at the left, that has experienced an average of almost 3.3 Celsius degrees in the last 10 years, and with an important loss of Antarctic sea ice, almost in the same region that we recognize as the most warming place in the Antarctic. However, this continent have another important issue regarding this particularity. In this place that was originally named as the Antarctic Paradise for sponge, or sponge paradise, because sponges, a benthic animal, occupy up to 80% of the available benthic surface in the Antarctic benthos. That also reflect that there has been described more than 6,000 taxonomically described species of Antarctica. So these animals have an enormous diversity in terms of taxonomic richness that are abundant in all benthic habitats and with extremely rich repertoire of chemical compounds. These animals represent the most ancient animal phylum arising more than 600 million years ago. So another particularity is that microorganisms associated to sponges can be up to 60% of the animal biomass. And most of the important biogeochemical process that support sponge for the Antarctic ecosystem relies on these microorganisms' activities. Here I show you what we have understood in the last 15 years. Our approach normally tries to integrate not only the bacteria and archaea component of the microbiome from Antarctic sponge, but also the eukaryotic component. So we try to look at a three-domain microbial diversity on these holobionts, defining holobionts as the organismal unit that integrate the sponge, in this case the host, the animal, with all the microbiota that accompany the sponge. And we can see that at first insight, bacteria and archaea and eukarya have completely different patterns of diversity and shareability between sponges and the seawater. I must remember you that sponges can filter seawater, that is their mode of nutrition. They incorporate carbon and other nutrients directly from the environment. And we can see that bacteria archaea are only shared between the seawater and the animal in a 10% of the total amount of species or ASVs or OTUs that we can look for, while in eukarya they share 66%, and the percentage that is exclusive from sponges in the case of the microbial eukaryotes, it's just 15% in comparison with an almost 80% that is unique from the bacteria. That means that the interaction between a bacteria archaea with the sponges and between eukarya and the sponges is completely different. We also detected several groups. In the case of bacteria, most of the microbial composition is almost exclusively described by gamma protobacteria, alpha protobacteria, but also Bacteroidea and Verrucomicrobia between other groups. In the case of the eukaryotic microbial composition, we detected that most of the more abundant groups are ochrophyte, that means diatoms that are a particularly important component, a photosynthetic component of the microbiome, but also an important part of dinoflagellates. Dinoflagellates are a kind of microorganisms that can have several modes of nutrition, so have a very broad diversity of nutritional modes. So we focus a little bit on what happened with the Antarctic sponge and the dinoflagellates. what we could see if we compare 3 different summers from sponges taken from the Antarctic Peninsula is that in comparison with sponges and seawater, sponges are dominated by a group of dinoflagellates that are not photosynthetic. They are in fact members of the protozooplankton, and you can see in the last part of this graph that parasites are in fact most abundant in the sponges compared to the seawater. We also try to understand if these signatures in microbial composition are particular from Antarctica or are generalized, and we include an analysis with all our Antarctic sponge samples, and we include this with the ones that are deposited in the global sponge microbiome, a global effort that have several samples from tropical and temperate environment, and we detect that in fact our Antarctic sponge are completely different in terms of microbial composition with all the sponge from other environment. Surprisingly, our sponge, Antarctic sponge, also harbor more positive interaction between bacteria than between Eukarya and Archaea and the sponges. The most representative groups that are interacting in the Antarctic sponge olobionts are the most abundant one: gamma protobacteria, alpha protobacteria, and bacteroidea. And we also detected that in comparison to seawater, Antarctic sponge have an enriched in genes related to symbiotic lifestyles. All these genes are increased, even though they are present in the seawater that you can see here in green, almost blue. But in the 2 sponges that we start with metagenomic analysis, we realized that CRISPR-Cas, toxin antitoxin, restriction modification, mobile genetic elements, and eukaryotic-like proteins were highly abundant. Other things that we detected is that Antarctic sponge microbiome have 4 of the 6 described metabolic pathway for autotrophic carbon fixation. This is the more broad repertoire of for carbon fixation that we detect for sponge microbiome. So that means that some of the functionality of this sponge microbiome could in fact reflect the supply of carbon and other photosynthetates to the host. We also detected or identified some MACs. In this study, we collect almost 30 MACs from Antarctic sponges that belong to gamma, alpha, some archaea, some groups. We detected signals of symbiosis like genome size reduction, GC content reduction in some of the beans. And we also detected a metabolic repertoire that was reflected in the metagenomes assembled for genomes that can be identified related to carbon fixation, for example, RBCyl genes, Woodhull-Lundial pathway, hydroxypropionate, reverse tricarboxylic cycle, ammonia oxidation, denitrification, degradation of urea, several genes for sulfate reduction or biogeochemical cycling of sulfates, vitamin production, a lot of genes related to symbiosis, and that what finally surprised us more is that in all our MACs, we detected a lot of genes related to cold adaptation. So overall, our study from the last years in Antarctica have revealed several patterns that we recognize as unique from this ecosystem. First of all, that Antarctic sponge microbiome are not similar or are not the same as the sponge microbiome from other environments. In Antarctic olobionts, we have a particular compositional and functional signature. that positive bacterial interaction in the Antarctic sponge holobionts prevail, while stable parasitic dinoflagellates are important constituent of the Antarctic sponge holobionts, that functions related to symbiotic lifestyle, as expected, I must say, are increased in sponge microbiome, and finally, that the unique metabolic potential of Antarctic sponge microbiome, we think that could help the sponge holobiont to survive in this environment, and is part of the reason why sponges or sponge holobiont are so abundant and important in the Antarctic benthic ecosystem. So thanks, this is the group and all the funding, and thank you very much.